I will give a brief introduction to CassavaBase. CassavaBase implements a complete digital ecosystem in which data is always collected and processed in a digital form. The goal is to increase data quality, make data sharing easier, enable advanced statistical analysis over large data sets, and to create a repository of breeding data. The CassavaBase digital ecosystem covers the entire breeding cycle, including germplasm management, pedigrees, trial management, phenotype collection and evaluation, collection and management of genotyping data, as well as data QC, analysis, and selection support. CassavaBase is completely web-based and is running in the cloud, so no software is required other than a web browser. All data are stored on the CassavaBase servers. It is accessible at https://cassavabase.org. A brief overview of the user interface. Every page has a toolbar. The search menu gives access to all the different searches, with searches for accessions, trials, genotyping information, traits, etc. The Manage menu gives access to all the breeding management functions, such as managing trials, crosses, genotyping plates, barcodes, and so forth. The Analyze menu gives access to all the different analysis tools on the site, including SolGS, the genomic selection tool, clustering, PCA, GWAS, stability and heritability tools, and others. The About menu, importantly, gives access to the CassavaBase manual and instructional videos on YouTube. To use CassavaBase, you first have to log in and to create an account. Each user has assigned roles in the database. The roles decide what a user can do on the website. Different roles exist for reading data, adding and deleting data, and other actions. CassavaBase is used by a worldwide community of cassava breeders, with a total of 24 breeding programs registered in the database. This map shows the trial locations throughout the world. A clear focus can be seen on Africa, which has by far the most activity, followed by South America and Asia. The major driving force behind CassavaBase is the NextGen project, for which the database was initially created. CassavaBase contains huge amounts of breeding data, and some programs, such as IITA and CIAT, have uploaded historical data dating back to the 1970s. Currently, CassavaBase contains almost 5,500 trials, 20 million phenotypic measurements, and almost 500,000 genotyping results. An active breeding program, all data is managed from within the database. For example, crosses can be planned and recorded from within the database, and trials can be designed and progeny performance predicted. This slide shows an animation of a so-called workflow for trial creation. The workflow requires some parameters to be input, such as the name of the trial, the type of randomization, and the list of accessions to be used, and then generates a trial and stores it in the database. A trial detail page is available for every trial in the database. It shows basic information about the trial, such as the trial name, location, and trial layout. Easy to access tools can calculate ANOVA, heritability, or clustering. The field layout and summary of the data collected is also shown. These are the analysis tools, and there's a lot more sections. We have recently made a lot of improvements to the field viewer, which is now more flexible. It allows to set the origin of the trial as well as border rows and can determine planting and harvesting order. Once a trial has been created, it can be phenotyped using a number of tools. The most important to date has been the Fieldbook App that allows data to be recorded digitally in the field. The Fieldbook App runs on a tablet that can be taken to the field and data can be collected and then uploaded directly to CassavaBase. The field app was created at the Kansas State University. To enable easy identification of plant accessions in the field, CassavaBase supports the barcoding of fields. CassavaBase has an integrated label designer tool that allows users to design custom labels and print them right from the database. The labels can be read by the tablets running the Android Fieldbook, greatly reducing data error. Portable NIRS devices are revolutionizing phenotyping. Spectra can be correlated with traits in samples. The calibration of this trait spectrum correlation can be performed in CassavaBase. Nick will present more about this later in the workshop. CassavaBase also has a module called ImageBreed for drone-based image analysis. Drones take pictures while flying over a field. These images can be assembled in CassavaBase and overlaid with georeferenced plot coordinates. Plot-specific images can be excised from the different wavelengths and then later analyzed. For a root crop, being able to non-destructively sample the root system is of key importance, and we are working with IITA and others to implement ground-penetrating radar analysis for this purpose. CassavaBase also implements a full genotype database for low to high density genotyping methods such as Intertech, DartSeq, and GBS. It is extremely important to collect the data using standard operating procedures such that genotyped samples can be matched with accessions and samples in the database. The SOPs rely on the Coordinate app, which is used to collect the sample data in the field and group it into 96 well plates. The information is uploaded to CassavaBase, from where it can be submitted to the sequencing centers. The resulting genotypes can then be processed and uploaded as well and used for analysis, such as SolGS in CassavaBase. Last but not least, the data in the database can be queried. We have seen a lot of the searches in the search menu, but there is also a special query tool called the Wizard. Data in the database can be considered as an n-dimensional cube with the different dimensions represented by the factors in the data, such as breeding locations, years, breeding programs, traits, and so forth. One way to query the data is to conceptually slice the cube along the different dimensions. The result of the query will be an intersect. With this approach, very specific data can be generated for further analysis. This sounds probably more complicated than it is. This concept is implemented in the wizard, which displays 4 columns for each of which a dimension can be selected. For every dimension, the data in the database are displayed and can be selected. For example, the breeding programs, as here in the example. Next, the years are selected that have data associated with the breeding programs, and then the locations that are used by the breeding programs in those years, and finally the traits are selected. These selections can be stored in the database and then be fed into the tools for further analysis. I'd also like to mention that CassavaBase and all breed-based databases implement the full BRAPI 2.0 specification. This allows many tools that use BRAPI, so-called BRAPs, to access the data in the database directly. There is also a special package for R, BRAPI-R, that can access CassavaBase data directly from within R. Future work. We are constantly working on improving CassavaBase. Improving the existing features as well as adding new ones. Currently, some of the things we are working on include what we call high dimensionality phenotypes, which includes the NIRS data, but also transcript data, proteomics data, and ground-penetrating radar data that we have already mentioned. We also think that BRAPI is one of the most important approaches to standardization for breeding software, so we continue to work with Peter Selby to evolve the specification as well as our implementation. In that context, we are also BRAPifying, if that's a word, many of the tools on CassavaBase to make them more widely useful. Finally, we are working to close some of the gaps in processing of genotypes, adding standard workflows for SNP calling and imputation, and adding support for polyploids. I'd like to acknowledge all the lab members as well as all the breeders, data managers, and breeding staff in the breeding centers that have made CassavaBase possible. Thank you.